Good morning, everyone. Welcome to the June 2025 Zoom Citrus Seminar. Thank you for joining us. First, I would like to thank Alison Walton from the HLB Center for her help and cooperation. Today's program offers one CEU for pesticide license renewal and one CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. Today's program will focus on current progress in improving citrus resistance against HLB via CRISPR genome editing and other horticulture approaches. This is one of the most important strategies to come up with a cure for HLB. Our guest speaker is Dr. Nian Wang, Eminent scholar, Endowed chair, and a professor in biotechnology at the UF-IFAS Citrus Research and Education Center in Lake Alfred. Dr. Wang will be covering current progress in genome editing of HLB susceptibility genes in citrus to improve disease resistance, and will go over field trial data of testing different horticulture approaches to control HLB. Dr. Wang, it is all yours. Please go ahead.
Good morning. Thank you, Mongi, for the invitation. Thanks, Alison, for the help. Today also invite Dr. Dawson and my colleague to join this my presentation. So he will provide wisdom for some relevant questions. So today I'm going to mainly talk about two trees we have been working on in the past. Let's see, I joined here since 2007. I see more than like almost 18 years. Yeah. Okay. We have I want to see it has been a long process. We have journeyed we have developed the CRISPR gene editing technology from the from the scratch. We have identified putative genes to edit, right, to improve disease resistance against HRV. So overall, we have generated more than 200 lines, focusing on 40 target genes. We mainly focus on the two areas. One is suppressing HRV symptoms. Another one try to kill CLAS. Today, instead of talking all of them, I only focus on the two. They are the two, the two target genes, genetic line for those two target genes that are ready for you to use. So our first batch of non-transgenic genome-edited Valencia and Hamlin through Orange have received regulatory approval by HLB and EPA. This is for the first target gene, so-called EDS1. So here are some representative plant. You see here the EDS1 edited Hamlin. Here are EDS1 edited Valencia. They are still pretty small, but they are getting, I think they are getting ready for propagation, especially the Hamlin lines. Valencia is still relatively small. They say I take last week, so I think it should be ready for the propagation pretty soon by nurseries. Let's give you some timeline regarding what we have done for the regulatory approval. So we filed the HLB approval on April 24th. We received first response back on April 30th. We submitted revision on the same day. We received the basically received the regulatory approval on May 1st. So in terms of EPA approval, it was submitted on April 24th. We also received the first response back on May 1st. Then we submitted a revision on May 21st and approved, and was finally approved by EPA on June 4th. So basically, in terms of if we want to use those plant, we have received a federal approval for you can use them. I also want to give a shout out for the IFAS and EPA. If you can, please send them, your praise to them. They are the best federal agencies I have worked with. They care about the industry so much. They really want to move forward with this plant as soon as possible. We also need another approval at the state level. It's the DPI, New Biotechnology Program. I think Justin and Idong is the one of the best, and his team have give us the best support, so we can move the plant to nursery for propagation through a fast track. If you have not heard from the plant have not been get approved by the state, please give Justin, your push. So I want to tell you why we think this plant are good. So I think to work with HLB to generate to improve data returns against HLB, you need to make sure you have a suitable target genes. In situ, right? That's more than 30,000 genes. It's impossible you work on all of them, right? So make sure you really have the right gene you can work on, right? We select 40 genes. It's naive to all of them will work, but I think at least today the 2 I think should work. You do the conduct CRISPR gene editing of those 2 genes. We think will improve either tolerance or against HLB. So the point is, right, if you not only have HLB rating and tolerance, right, the plant should still able to produce good quality of citrus fruit. You should not have a significant effect on the plant growth or anything. Or you cannot only get the returns and tolerance without the fruit, right? Because it's a long-term, juvenility for citrus, we need to generate biallele or homozygous mutant in the T0 generation. Okay. Make sure our CRISPR gene editing technology is very specific and no off-target mutations. And also, right, this should be hopefully this will be non-transgenic and non-GMO. Because based on our experience with citrus cancer in gene editing, we generate our transgenic cancer return trace in 2020. But then it's almost impossible to go through the federal regulation pathway. It's just so hard. Anyway, and another point is, right, if you can bypass the juvenility if possible, right? Because if we had a plant, we hope you can plant them and get a fruit as soon as possible, right? So I want to give you more information regarding why we think the EDS1 is a right target to work with. In 2023, right, 2022, we found the citrus HLB is a pathogen-triggered immune disease. So this is why do we say that, right? Basically, if you have heard about many talks by colleagues in CREC and from UF, right? You see HLB when they infecting citrus, right? So they trigger the reactive oxygen species. We simply call it ROS production and the phenol callus deposition, right? You see here, here's the wild type phenol cells. You see here the functional phenol cell element. But if in the cell are infected, right, you see the cell element really blocked by the callus, right? You see a lot of the phenol cells actually really dying, right? And here's the higher level of RS level in Silas HLB infected plant, right? He's a healthy one. This is HLB plant. Okay. So those, RS production and the color deposition, they are typical immune response. Okay. However, we don't have a genetic evidence for this disease model. That means we don't know what are the suitable target gene to target, right? So I want to go through. We have made some significant progress in terms of how Silas cause the HRB immune response and immune disease. Okay, we found out. We just give you some very make a long story short, right? We see when Silas infecting plant, they cause our production directly in the chloroplast. Okay, and also they cause the phloem callus deposition. The phloem color, RS we know, right? RS when they have a high amount of RS production, they can kill phloem cells. Okay. We also found phloem callus deposition can also lead to phloem cell death. Basically, right? That means RS production and the phloem cell callus deposition, they both can cause phloem cell death, and they both contribute to HRB symptoms. So if you only manipulate either RS production alone or the phloem callus deposition alone, you can only partially reduce HLB symptoms or damages. That means, right, you need to find this master regulator, okay, that can control both. Okay, so we have been looking for the gene that can control this the Silas triggered immune response. Okay, so it's actually a very complex pathway, right? Many genes actually involved. We just look here. I'm showing you a very simple figure that shows you actually the most critical immune signaling genes. Here I only focus on EDS1. Here you see this is central here. Pedophile. We also have mutant for SEG1, also BRK1, right? Sub1, and also RBHB. So those genes are really critical genes involved in immune signaling. Okay. We have a genetic mutant for citrus, but many of them still under inoculation. Citrus, they take a long time to develop symptoms. So meanwhile, we work with several collaborate with several colleagues from Cornell, from UC Riverside, from Texas A&M, and from James Madison University to test the tomato library. Why we do that? Because liver vector not only infecting citrus, they can also infecting tomato. You see, right? We see, right, when Silas infecting citrus, it causes RS production and phloem callus deposition, right? So actually, when you see liver vector infecting tomato, you see this trypan blue staining. This means cell death. You see, right? They also cause the cell death in tomato. They also can cause phloem callus deposition. You see here, white type, right? It's here the healthy one. You see a functional stable element, but you see when the liver vector infecting, right? They basically cause also callus deposition. And similarly, you can observe liver vector infecting tomato cause the RS production accumulation as well. So basically, we can use this as a model, right? Because tomato is a much faster system. We can finish the evaluation within months. Okay. Yeah. So right, we have a here's the pedophile mutant. Here's the EDS1 mutant. Here's the RBHB mutant, actually. Here's the sub1 mutant and the BRQ1 mutant. You see here, again, right? You see here, for the Y type, when infected by Zekri, by liver bacteria, right? They die very quickly. But if you knock out the genetic editing of EDS1 and the PAD4, right? Basically, you see, they basically abolish the disease development. Okay, right? And also the EDS1 mutant, they reduce the RS production. They reduce the ZS. You see here, right? Is the ZS in the wild type, but in the EDS1 mutant, you see the ZS much reduced here also as well, and also they reduce RS production, and also they prevent the color deposition induced by liver bacteria. You see here, right? Either in the wild type, the phloem is totally blocked, but EDS1 is not blocked, right? So this give us, right? This gives us some like a hint, right? Basically, EDS1 and the PAD4 will likely to be excellent target to suppress the disease symptom caused by liver bacteria, especially SILAS, right? So now we have generated use the RMP method. We basically do the single cell gene editing. We did then we do the 6-month regeneration. You see some of the plant. We have did the inoculation with liver bacteria SILAS on May 15th through SILAS transformation. You see here. Here's the EDS1 edited Hamlin, the wild type Hamlin. Okay. So why do I think the non-transgenic EDS1 edited sweet orange are potentially good choice for you against HRB? I think based on our current understanding is if you knock out EDS1, they will abolish SILAS induction of RS production and phloem color deposition and the phloem cell death. So the plant will likely to be HRB tolerant. So you will not affecting SILAS population, but they will basically turn the SILAS into a endophyte, right? There's not the damaging endophyte, like the microbe in the gut. Okay. Okay, I think those also those plants have received HLB and EPA approval, so you can use them if you want. So based on our I see your plant, right? The plant has no difference in terms of their look, in terms of this is not affecting the fruit quality based on our previous knowledge on the EDS1. Okay. So again, the plants are ready for you for planting. Of course, we need a propagation by nursery. And also, again, why it's important? Why it's a good target, right? The recent science paper, right? They identify PAP21 is it's a susceptibility gene for switch HRB. Actually, the PAP21 is in the same pathway as EDS1. You see, EDS1 actually positively regulate the PAP21. If when liver bacteria infecting plant, they induce PAP21. They cause all the major they contribute to disease development. But if you knock out EDS1, right, the induction of PAP21 is significantly reduced. So that's why maybe this explain why EDS1 will work. Okay. Of course, we are also working on genome editing of PAP21. Actually, the plant we did the genome editing of 21 2 months ago. I think we should get a plant of PAP21 edit plant sometime this year, I think. So we need to have some caution. You got EDS1. It's not perfect. But also how do we address this problem? So muted EDS1 why likely reduce accumulation of salicylic acid. You know, SA, we call it SA, right? SA is an important defense hormone. So they might reduce the resistance against some biotrophic pathogens such as cystic cancer, but they will likely increase resistance against Phytophthora. You know, that's based on what we know about the EDS1 mutant study in other crops. Okay, so how do we address this issue, right? With this, is that really detrimental issue, right? I think it's not. I think the benefit of using EDS1 edited plant actually way outweigh the cost. You know why? There's several reasons. Okay. Right? When we plant sushi, right? We know we have the Sayang, we have the rootstock. Rootstock, most rootstock are, basically rootstock were not edited for EDS1, right? So this issue for the rootstock is void, right? Okay. And also, because the main compromise comes from the reduced accumulation of ICA, right? So you can, we do the spray lot by the seed growers. So basically, you want to do the foliar spray with SA once in a while. So you can really, how to say, try to remove that compromise of the ICA reduction, right? And also, I think HLB is really killing the trees. Other disease in Florida, I think we all have pretty efficient control measurement. So I hope this plant, EDS1 plant, will have us solve this HRB issue first. Okay. Okay. Another important issue, right? Juvenility. This plant we use basically go through the single cell genome editing and then do the regeneration. This basically considers juvenile tissue. They need to go through the juvenility before they produce the fruit, right? If you know Southern garden, right? The transgenic plant, right? They take a long time. They still did not generate fruit, right? So how we can let the non-transgenic EDS1 edit the sweet orange, harmaline, and valentia to fruit quickly? Okay. That's something I think we need to work together. Okay, growers need to maximize the growth to shorten the juvenility. Here's the previous study already showed the most promising treatment to shorten or change from the juvenility to mature is to grow seedlings as fast as possible to a certain minimum size, and then if necessary plant them in flowering inducing condition. Okay. Another one, there's something we have been working on breeding, right? Actually, I found this, I think there's one way you might shorten this juvenility. When we work with breeding, we often use, put the juvenile tissue on the mature trees. We basically call it top working. Top working, right? Means basically the bottom part is the mature trees, right? You put the juvenile tissue on it, so the plant, that juvenile tissue will flower very quickly, allow it to do the study. That's I sound okay. If that work, right, we can just simply use similar principle, just design a little different trees, right? Now how we do that, right? Because when the juvenile, right, where the flowering molecule from? The flowering molecule come from the mature leaves, right? From the mature leaves, right? Mature leaves, remember, right? So they can transport between the tissues, between the branches, okay? That's how the top working work, okay? So what do I did, for this one, what I mean by design trees? So when you buy trees from nursery, right? Normally that's what you do. You see, you put this mature tissue bark on the rootstock, right? either UF-942, either or UF-5, either like this, right? Because this normally the tree from the nursery, right? This already bypass the no juvenility issue. They're mature tissue, right? Okay. What you need to do is for here, simply I think you can do the instead of one bud, you put one bud from the mature tissue, right? Traditional bud, no juvenility. Another bud from the genome edited EDS1 plant. Okay. So you need to put instead of one bud, you put two buds. You see here, simply this is the cyan mature tissue, the cyan EDS1 edited, right? Because this plant has two. Another way is you can do is you bud this EDS1 on the mature branch. Okay. So let me tell you why it works. Why it might work. Because right, the EDS1, because the mature tissue, this branch will will provide the flowering signal molecule to the EDS1 juvenile tissue. Okay. So they will shorten the juvenile tissue stage for our EDS1 edited branch. So another benefit is the EDS1 edited branch will provide a nutrient to root to prevent tree death, to keep the wild type branch healthy and more productive. The key point, right? This plant is actually composite tree, right? We think you have wild type Hamlin or Valencia and the EDS1 Hamlin and the Valencia. Okay. I think those two, those because only one single gene editing, I assume there's no difference in terms of fruit quality, because previous study EDS1 mutation have no effect on fruit quality. Okay. So I think that means, right, you do can like so it does not matter as long you keep the wild type fruit, produce fruit, right? Tree produce fruit, branch produce fruit. That's something we can that work, right? Let me tell you why this what I mean. The EDS1 edited branch can keep the wild type branch alive. Okay. So let me to answer this question. When you study how does HLB cause tree death. We know liver vector infecting citrus, right? They cause phloem cell death, the and the RS production and the phloem color deposition in the tree, right? Actually, we found that only happen in the photosynthetic tissues, like mostly in the leaf, in the trunk, but those things actually not happen in the root. Okay. So how does Silas cause if you see Dr. Jim Graham study, right? You see when Silas infecting citrus, right? They cause the root death. So how does the citrus cause Silas cause the citrus root death? We found actually, you see, if you really observe, if you see the root cells at a different stage, what we did is we remove the root and then we let the new root grows. Then we look at the root death at a different stage weekly. Okay, you see, we see at the 1 week to 4 weeks, right, to 5 weeks. You don't see phloem cell death, but at week 5, you begin to see root cell death. But root cell death, this is very universally, not only focus on the phloem, not like when Silas infecting leaf. That's mostly in the phloem cells. So especially if you look at the starch, right? Starch is the nutrient for the root. You see the dramatic reduction of starch in the root. So and also we also look at some root from different cyan, and they all on the UF-942 rootstock. You see, even the root have has no Silas infection, but they all have similar root death. So what I mean here, that means I think the CLAS does not really cause root death directly. Actually, it causes because the nutrition, the nutrient starvation, because when CLAS infecting the photosynthetic tissues, they cause the root cell, RS production, cause the phloem cell calorie deposition, then cause the phloem cell death, that block the nutrition transportation to the root, right? Then the root die, then contribute to the tree death. Okay. That means, right? That means if you provide enough nutrient to the root, you can prevent tree death. That's what I mean here. So the EDS1 edited branch will be able to provide the nutrient to root to prevent tree from dying. Okay. So let's see how the let's give imagine what will happen, right? So normally by year 3, the mature branches from the traditional grafting, right? They can produce fruit. Okay. But I assume this EDS1 edited branch still not producing fruit. But I think in year 4 into year 7, the cyan branch from the mature tissue will normally generate fruit and more and more fruit, and then EDS1 edit line, they will be able to overcome juvenility little by little because the flowering signaling from here to here to the EDS1 branch. Okay, they can be getting fruit. So by year 8 and after, so both white type cyan and the EDS1 edited cyan will produce fruit. Yeah. So by the year 8, right? If you want to plant the EDS1 pure EDS1 edit lines, you can. They should be available to you as well. Okay. Okay. Then if you have some trees in the field, if you want to replace them, actually you can simply do a top working. But I think I don't know. Let's assume this one will take a lot of labor. So, but anyway, if you want to do this, is at least in top working have been known to really shorten the juvenility. Okay. Actually, this is a slide that I steal from Mongi. So nice picture from Mongi, our host. And also another way to overcome the juvenility is use the CTV vector. This is developed by Dr. Chova and also Dr. Bill Dawson. Dr. Dawson is also in the audience. So this one, I think you probably have multiple ways to use them. I think one way I think is probably when the tree reaches a certain minimum size, you can inoculate the FT3. This is a CDV vector. You can grow quickly if this vector has been approved by if it's an EPA. Okay. Next I'm gonna talk about second plant. Okay. My second plant is it's also non-transgenic edited. This is the gene target DMR6. DMR6 is the broad range disease susceptibility gene. What does that mean? That means actually this is disease range genes against many diseases. If you knock it out, you increase disease returns against many diseases. I will explain that later. Okay. So here's the Hamlin. Here this DMR6 edited Hamlin. Here the DMR6 edited Valencia. So we have filed the regulatory approval by IFAS on June the 2nd. So we submitted a revision on June the 8th. We filed the EPA approval on June the 9th. We have not heard back from them. Well, hopefully we can receive the regulatory approval pretty soon. So why do I think the non-transgenic DMR6 edited Sweet Orange are potentially a good choice for you? So what does DMR6 do? DMR6 is right here. You see, assay, we know, right? The assay is actually really important defense hormone. DMR6 actually degrade SA, so you can reduce SA accumulation in a plant. Okay. What do we do is, if you do the genome editing of DMR6, you increase SA level inside the plant. So basically, that's why it confers resistance to many different pathogens. Again, right? DMR6 will increase this resistance against the biotrophic pathogen. Yeah, but not necessarily against natural pathogen. It's a little bit different from EDS1. Okay, so based on the previous study, we think that the genome editing of DMR6 will enhance the killing effect of the plant defense against the SILAS. So basically will reduce SILAS population in plant. Okay, let's see again, right? Let's see, I see how does the assay work, why assay is important. Defense hormones, you see, that's the reverse relationship between assay level and plant growth. Here is the assay is involving the resistance against biotrophic pathogen, right? That's why you increase the assay level, you likely increase the resistance against SILAS. And also, in the past study, if you knock out DMR6, you see the increase the resistance against Pseudomonas syringae, Xanthomonas gardneri, Xanthomonas perforans, and also Xanthomonas campestris and Xanthomonas citri. You see, this DMR6 is pretty. They have pretty good. If you knock it out, right, you have pretty good effect against many different pathogens. So what's going to happen for the next step for the non-transgenic DMR6 edited Valencia and Hamelin? So we have done the inoculation of the non-transgenic DMR6 edited lines. So right, we submitted the paperwork for regulatory approval by HLB and EPA. We need to work with the DPI, New Biotechnology Program. We needed to do small scale field trial. We needed to work with nursery for the propagation. This one also have a similar issue in terms of the juvenility, right? So I think the composite design I talked earlier for EDS1 will also can be used for DMR6 edit line. So timeline for the non-transgenic EDS1 and DMR6 edited citrus plant to be used by flower citrus growers, right? You see, in 2005, we already finished the paperwork, regular approval for EDS1 by HLB and the EPA. So we are doing this approval is ongoing for DMR6 edit lines. The HLB testing, we have done the inoculation. I think normally within 3 or 4 months, you have some good idea regarding the plant will be good or not. But in terms of solid data, normally you need to wait 8 or 9 months. Yeah, right. So we need to work with DPI, New Biotechnology Program. We need to work with the nursery for the propagation. I think how the tree will be available to the growers. I think it unlikely be available to grow this year, but in 2026, 2027, I think if you are interested, there'll be small scale trees. You can do small scale trees in your grow to do field trial, because they have received HLB and EPA approval. Basically you can test them at your will. I think the trade will be available to you at large scale in 2028. And okay, the question, right? So how we can work together to speed up the delivery of the trade? We we have 40 target genes. Those two genes, today I'm talking about those two, because I think those two gene targets are the best target we can have. They should work well against HLB. Of course, I think we need to have solid data to see that. But if I bet, I'm gonna bet those will work. So if this work, I think as a scientist, and as a UF scientist, I think we did our part to give you the tree. I think as growers, you need to step up the effort to get them approved by Florida DPI, New Biodiversity Program, and plant them. Okay. Okay, maybe this I'm thinking, maybe other bottleneck you can think of. I don't know any colleague are in the nursery. So if you're interested, you can let me know as well. So take home message. So the non-transgenic EDS1 edited Hamlin and Valencia are ready for Florida city growers. The non-transgenic DMR6 edited Hamlin and Valencia also almost ready for the city growers as well. So more genetic switches, about more than 20 lines for 40 genes are being evaluated. But I don't think those other targets will be better than EDS1 and DMR6. Or they should maybe some are similar, or including the PUB21, right? So I think the new tree design can be used to overcome the juvenility issue. Okay, with that, I thank my collaborators, the funding support. Especially, I want to really thank the ECDRE program, NIFA, USDA, NIFA, CDRE for their support, and the CRDF, Department of Citrus, for their support, because their investment to really help us speed up the process, because we bought so many group chambers that make this work possible. With that, I'm ready to take your questions.
Thank you, Dr. Wang.